Howdy, Eggs. My name is Mike Resendez, and today I've prepared for you a lesson regarding gene editing in meat animals. Uh, to me, this is an extremely fascinating and interesting topic. I was first introduced to it when I worked for a livestock reproduction company called Transova. Transova has a very, very small gene editing department. I essentially had no involvement in that. This is where I learned about gene editing and what the technology actually was. Through some research, I've certainly strengthened my understanding of it, but it's just a fascinating topic. And if it's something that doesn't necessarily jump off the page to you as something you're just so interested in or extremely passionate about, that's certainly okay. I think there's plenty of relevance in terms of gene editing when we talk about efficiency of animal and just animal welfare, especially when we talk about feeding a growing population. The numbers talked about like 2050 needing to feed a huge population with less and less space than what we're used to. Throughout this, we'll connect the idea of gene editing in meat animals to a lot of the concepts discussed by Rogers in his book, The Diffusion of Innovations. This is a picture we'll talk about here in a couple slides. I think it's extremely interesting and something that's really, really cool to me. With that, we'll get right into it and begin our lesson. Our learning objectives for today. And I want you all to think about this stuff as we get through this. This can be maybe a complex technology. And I think if we really, really hone in on our learning objectives, they're designed for a reason and designed to give you a path of what you're supposed to take away from this. Students who listen will be able to describe the relative advantages and disadvantages of gene editing in meat animals and how these will affect the rate of diffusion for this innovation. Students will be able to identify all five phases of the innovative decision process per Rogers. Students will be able to choose if they like or dislike the concept of gene editing while being in the persuasion stage of the innovative decision process. I always like when we start with an agenda or an overview, and these are the main questions that will be answered. And so we'll go over this. What is gene editing and how is it used in the meat animal industry? What are the pros and cons of gene editing, and how will this affect the rate of diffusion per Rogers? What is the innovative decision process per Rogers, and how does it pertain to gene editing? Followed by this slide, we'll have a quick pop quiz, and I ask that everybody follow along on their handout. It's a good engagement tool to make sure you're following along and then ultimately accomplishing our learning objectives throughout the presentation. So, in the broadest sense of the term, gene editing is the ability to change the specific makeup of DNA sequencing. Uh, Scientists and researchers have the ability to change one or multiple strands of DNA to affect the expression of a gene. Probably the most popular talked about ones are an animal that should genetically have horns, being gene edited to be smooth polled. This is often talked about from a safety perspective when we think about ranchers working cattle on a regular basis. And then another popular one is cattle that are genetically black hided to be changed to red hided. This is very important when we talk about heat tolerance, and the Bryan College Station is a perfect region where heat tolerance can be an issue in cattle. And so, just changing a couple of things in order to make animals more efficient is the broadest sense of gene editing. A quick history lesson on gene editing. In 1950, the double helix structure of DNA was discovered. And for most of us that have probably taken a basic science class in high school or college, this seems like a relatively general concept, something that's easily conveyed. This is groundbreaking when we think about just understanding the structure of DNA and how genes are expressed. And it ultimately led to technologies being developed in order to change these genes and splice them together. From there, we fast forward, about 70 years or so, or just over 70 years, where the CRISPR-Cas9 technology was discovered. It was discovered by Jennifer Doudna and Emmanuel Carpentier. And basically, what this technology is, it's the ability to microscopically cut a DNA strand, splice a new DNA strand in order to affect what genes are being expressed. And so this is what has ultimately propelled all the following research and stuff to be done with gene editing, not only in meat animals, but in humans as well. 2015 is probably the milestone that jumps off the page just the most to me personally. The first gene edited food animal was approved by the FDA to enter the food chain. This was the Aqua Advantage salmon. That was that picture we talked about on the first slide. where this salmon was edited to be, you know, disease resistance, pest resistant, and ultimately all that together made the salmon nearly twice as efficient when it came to converting, food to growth and being able to enter the food chain that much faster. Uh, Another big one is in 2020, the FDA approved the first gene edited pig to enter the food chain. And this was the Galsafe pig. Pigs have a particular sugar in their meats, or some pigs, uh, that many humans express allergic reaction to. So they gene edited these pigs to where they didn't express the sugar in their meat. And people that had this allergic reaction would then be able to consume pork without having an allergic reaction. And then another huge one, it happened last May, was Washington State University gets approval to use the first gene edited pig to make German sausage. Not only was this Washington State achieving their goal, and basically the whole goal of their project was to make various gene edits and then show consumers, meat consumers, that the product was essentially the same as it was without gene edited animals. Oftentimes a lot of people like to say gene editing is just speeding up, selective reproduction. And so that was Washington State's goal. And then outside of that, tons of land grant universities like Texas A&M and other ones have wanted to jump in on this research and try to get gene editing off the ground and be a front runner for this technology. That's a pretty quick timeline, especially when we think about last May. So it's still a relatively new technology, and just the 1950s, the initial part of the technology was developed. What is the commercially available top or technology of gene editing? Aceligid is probably the leader when we think about gene editing in meat animals, and they offer various technologies for swine, cattle, and fish. I want to point out not all of these technologies uh, offer where the animal can enter the food chain. This is what is commercially available primarily for research purposes, and I think Acelagen probably sees all these as ultimately beneficial to whatever industry they're targeting. And so these can be, various disease resistance, making animals more, just efficient when we talk about nutrition. One that's very, very interesting to me is the castration procedure prevention. So male pigs wouldn't end up dropping their testicles. They'd still be a male pig, but they wouldn't have to go through a castration surgery. Oftentimes, baby piglets experience a lot of stress when they have to get castrated, and it sets them back just a couple of weeks. When we think about them converting feed into pounds, that could eventually be pounds of meat entering the food chain. So this is one way that gene editing can make stuff more efficient. Another big one is improved heat tolerance. This technology is FDA approved, and this would be converting, a traditionally fluffy-haired animal to be slick-haired, or the example we talked about earlier, changing a red or a black-hided calf into a red-hided calf to make them, just more tolerant to the heat. I think it's very, very interesting that they have so many technologies that are available commercially, yet not all of them will allow the animals produced to enter the food chain. This is a growing industry, and there's just so many different things you can do with gene editing. So the pros and cons of gene editing, and before we get into this, I put pros and cons up here because that's probably the more often described term. Once we get past this slide, we're going to talk about relative advantages and disadvantages, which in my opinion are different than pros and cons. They're just a bit more specific. We'll start with reading Rogers' definition of relative advantages. And so a relative advantage is the degree to which an innovation is perceived as better than the idea it supersedes, or then we can infer a relative disadvantage is the way it's perceived as worse. So we're not just talking about gene editing and the things it does good and the things it does bad. We're actually making this, uh, you know, relevant to what we're talking about. So I think relative advantage is probably a better way to describe this. I just wanted to put pros and cons up there so we can be thinking, a bit more specific. So we're going to be comparing gene editing as opposed to genetic selection through reproduction here. And so some of those advantages or relative advantages are the ability to control what genes are expressed by an animal without generations of selective breeding. So that's exactly what we were talking about. These can affect animal husbandry and welfare. We talked about the slick hair versus fluffy haired or red versus black hided. Uh, there's the ability to make animals genetically resistant to diseases. And then another one that's huge is just making animals more efficient in terms of growth to reach their harvest weight faster. That's ultimately what we're going to talk about when we talk about ways to be more efficient. Is how can we get these live animals to their kill weight faster, and that'll drastically change the available amount of protein Americans and just citizens worldwide are offered. Has some relative disadvantages because not every innovation comes with only positives. It's heavily regulated by the FDA, and only certain gene edited animals are allowed to enter the food chain. We've talked about this with the coelagen already. Um, but yeah, what's the point of gene editing if we can't use it and not all the animals can enter the food chain? Uh, it's extremely relevant. This is a very, very relevant topic. It is a large initial investment. Gene editing is not as cheap as just turning a bull out with your cows or AIing some sows or making some fish. There's a large initial investment required to that. Um, some people and economists have described that the investment should pay off, but regardless, we have to have the capital and the overhead. And then gene editing is just extremely complex. And we think about Rogers' diffusion process complexity makes diffusing an innovation way more challenging because of how it's perceived by people. So, what is the current state of gene editing being diffused? And let me move my camera here so we can read our chart here. Before we get into just the state of gene editing and where it's at in the diffusion process, I want to explain this adoption curve that we have in the bottom right hand corner. So, the adoption curve is an S shaped curve that shows the typical trend for adopters over the lifetime of an innovation. And so obviously this can be as short or as long as it takes to fully implement an innovation or if it's successful. The trend line should show a spike between early adopters and laggards as we see reflected here with our orange S curve. And then when we think about gene editing and where it's at in its current state, we talked about acelagen having commercially available technology, but not all this technology is approved by the FDA. And then still, if we think back to our timeline, an extremely new concept here. I would very confidently say the gene editing in meat animals is in the very early stages of the innovators section right here. We haven't got to where 16% of people have adopted it, and we certainly haven't gotten past that to where 50% of people breeding cattle, pigs, and fish have adopted this technology yet. Some people hope it gets there, and if it helps feed the United States and the world, I'm certainly a proponent of that. I'm not the one to make the call, but if it has value and ultimately is diffused properly, I certainly can see this trend line taking place with gene editing. Next, we're going to talk about the innovative decision process and kind of switch gears to the back half of our presentation. As y'all can see, this is a linear process here. And we're going to go through and describe each of these here. So the first one is knowledge. Knowledge is not something that we spend time in. It's almost we're there and then we leave it real quick. So knowledge would be just simply hearing about the innovation, people seeking information about the innovation itself. So everybody that's listening to this lecture should be past the knowledge phase. You've learned a little bit about gene editing and meat animals, and then we move on. From there, we move on to the persuasion stage. And this is simply making a judgment call whether or not we like or dislike the gene editing technology. We're certainly seeking information still and trying to figure out exactly how it works and exactly how it could work. Um, from there, we move on to the decision process. And this is adopting or rejecting the technology as a whole. If we see its value and we like it, this is then we can move forward with it. And if we don't see its value or see there's too many risks involved with gene editing, we would then in the decision process choose to accept or reject this particular technology or adopt or reject. From there, we get into the implementation stage, and this is applying it to our lives or applying it to our uh, operations. So if we have done our research, we've learned about it, we've done our research, we've liked the idea of gene editing, and we think it makes financial sense and sense in the broader sense of the word. Then we then go ahead and implement gene editing and probably get a few animals gene edited. From there, we'd move on to the confirmation stage, and this is simply seeking reinforcement of the innovative decision. Naturally, humans want approval or want to know that they made the right or wrong decision, and this goes way deeper than just a personal sense, whether or not this was the right move for a business and something we'll continue to use. From there, I want to move on to a critical thinking activity. These are questions 7, 8, and 9 on your handout here. So we should have questions 1 through 6 already answered. And this is merely probably a personal answer to these questions. There's not necessarily a right or wrong answer. Uh, so where are you at in the innovative decision process relative to gene editing? Like we talked about, we should all be past the knowledge stage. So if we, we're an animal producer and we've done all our research, we can be in the decision phase or implementation phase. I would venture to say most of us are in the persuasion stage. Go ahead and jot that down on your handout. Next, we'll get into question 8. And as an animal producer, and if you're not an animal producer, I want you to pretend like you are. What will be your next step to take in the diffusion process of gene editing? This can be seeking more information. This can be implementing it, and there's multiple different ways to do this. This is simply an opinion on what you've thought of the technology so far. And then question 9 is more geared towards if we get back to and we decided we were in the persuasion stage. If you're past that, simply do not answer this question. But while in the persuasion stage of the innovation decision process, did you like or dislike the idea of gene editing? Regardless of your answer, I'd certainly like to see why you feel the way you do. From there, we'll get into a quick recap, and a group quiz here, just to make sure everybody was following along. So, when was the first CRISPR technology discovered? It's a multiple choice question. Question number 2: what 3 food animal species have the most readily available gene editing technology? It's something we talked about a lot with the coelagen and whatnot. Question number 3: a relative advantage of gene editing is that the technology is cheap to utilize. And then question number 4: list the 5 phases of the innovative decision process. Give you a few seconds here to finish your, jot this down or think about your answers. Then we'll go into answering our questions. So question number 1: when was the first CRISPR technology discovered? This was the Cas9 technology developed in 2012. Question number 2: the 3 food animal species that have the most readily available gene editing technology. That is cattle, swine, and fish. Question 3: a relative advantage of gene editing technology is that it is cheap to utilize. This is false. We talked about gene editing requiring a large initial investment. And then question number 4: the 5 phases of the innovative decision process. These were knowledge, persuasion, decision, implementation, and confirmation. Thank you all very, very much for listening. If you have any questions, please feel free to let me know. This is an extremely fascinating topic to me, and whether or not we move forward with it as an industry or not, I certainly think it's something that offers tons of relevance and something we can think about. Thank you all for listening, and I hope you have a great rest of your week.